Cool. Thank you so much for, can you hear me? Thank you so much for having me, and to the organizers of this symposium. It's my first time here in Ann Arbor, and it's nice here. I am a postdoc at UC Berkeley and UCSF, as Jill said, but today I'm going to talk about some of my PhD dissertation work, which is on bacterial fungal interactions, and specifically endosymbiotic bacteria that live inside of the hyphae of plant-associated fungi. So this is work that I did at Duke. When I was a grad student at Duke, Greg Benito, who's now at Michigan State, was a postdoc, and he was really influential as a mentor to me. So this is an ongoing collaboration between the two of us. It's off. This will work. Okay. So I'm going to start with a little overview of fungi in the rhizosphere. So we know that there are actually many microbes that live in the rhizosphere. It's probably a term that's accessible to us, but I'll remind you that it's just a technical term for the area directly around plant roots. So here what I'm showing you is a single root tip that's been colonized by a symbiotic fungus. And I've false colored this image so the fungal tissues are in red and the plant tissues are in purple inwards. And so you can see that these microbes wrap closely around plant roots, and what that does is creates an interface for resource exchange between the two. Now here I've illustrated a single fungus. In reality, there are many fungi, bacteria, and other microbes in this area, and their co-occurrence in regular patterns that we see with plants gives them an opportunity to interact and eventually to evolve into symbioses. So we know many examples of the direct interactions between plants, fungi, and bacteria, but what we know less about, and what this talk is really about, is the interaction between fungi and bacteria and how those interactions maybe scale up to above ground plant health. So I'm going to show you a few more trees in this talk, but they'll be phylogenetic trees. I study bacterial fungal interactions in vitro in lab. And really what I've been trying to do is use this system to ask a broader question that I'm interested in, and it's how do symbioses evolve? So for bacterial fungal interactions, specifically these long-term co-evolved endosymbionts that we've been thinking about, presumably once they were two free-living microbes. And I'm going to try to make the argument to you that as two free-living microbes, if you live alone, if you're two free-living individuals, life for you is really different, right? You have to respond to a lot more selective pressures like nutritional resources, competition, and those really change. So the selective landscape is different inside of eukaryotic cells. And you can see that actually written into the genome as a selection of symbiosis. And when symbioses move over towards this obligate end of the spectrum, the rate of genomic change actually sometimes increases. So what actually happens along this timeline? Well, most symbioses start with dinner. You got to be in the same place. You have to come into physical contact, decide what resources you're going to exchange and when, potentially why. And then as facultative symbioses move more towards obligate, what you have is a selection on what I call symbiotic genomic content. So oftentimes in bacterial fungal interactions, the resources they trade are metabolites, so potentially on the genes that are involved in the biosynthesis of these pathways. And in these metabolic exchanges, sometimes it's a wholesale, you make A and I'll make B. Other times it's a collaborative, I'll keep the gene that codes for the enzyme in step 1, you do 2, I'll do 3. What that does then is list the selective pressure to maintain the entire pathway, and it can lead to gene loss. And sometimes in long-term obligate symbioses like chloroplasts are a stunning example. You actually have genetic exchange. So the fungus then that I'm going to tell you about today is on this end of the spectrum in terms of obligate, and it's Mortariella elongata. So Mortariella is a robust producer of lipids. It's actually an industrial workhorse, and these lipids or fatty acids come in different flavors, including the polyunsaturated fatty acids like arachidonic acid. They're really important for the fungus basal physiology metabolism, but it's also a resource that's pretty rich, and you can use that as sort of a bartering chip for symbioses with, we think, plants and bacteria, and it's central to the story here. So Mortariella is a fungus in a group that we used to call zygomycetes. More recently, we're calling this subgroup Mucoromycota, and so the star of this story is in the red box there. But what you can see is that the pink and red branches that I've colored there, those all have endosymbionts that are closely related to each other. Now this is a phylogenomic tree, so we've used a lot of genes. We can have a high confidence in this topology. And one conclusion that we've derived from this tree is that close relatives of fungi harbor close relatives of endobacteria, and this speaks to the ancient nature of this relationship. So we can use data like these to date these trees, and we see that the divergence between the fungi that house related endosymbionts is around the same time as their bacteria. So this relationship is quite old. The endosymbiont itself's name is Mycoavidus. It's a sister to the genus Burkholderia, which is there on the pink branches. And then Glomeribacter, which is, see my arrow? It's there. That's another endosymbiont of a closely related fungus to Mortariella. So when we do see these endosymbionts in transmission electron micrographs, they're often clustered around lipid droplets. So I'll remind you that this fungus makes a lot of lipids. It's been challenging to image these bacteria. We found out later that they're really low abundance relative to their host, but when we do see them, they're always around these lipid droplets. And we think that's because they're using them directly as a nutrient resource. So we have genomes for both the fungi and the bacteria. We call this a twofer because when we sent out the genomic DNA for the fungus, we got back 2 genomes. So the fungi are growing on the plates, they've got the endobacteria inside, you extract DNA from the whole thing, and then you sequence and assemble. You can use fun metagenomic tricks I don't have time to talk about today, but if anybody wants to chat later, very interested, to assemble both genomes. And so we've done that, and we got with JGI and got some long packed bioreads involved and actually closed the bacterial genome. So that's what I'm showing you here. I've got genomic position along the outside, and then some of the genes on the various strands and some information about GC content and skew. And then on the middle, some functional genes. So in a lot of ways, this bacterial chromosome is like many other bacteria. But in one important way, it's very special and unique, and that's that this genome is highly reduced. So in the same way that I try to build logic in the intro for this idea that any obligate symbiosis started as 2 free-living organisms, we can gain insight into what happens along the way by comparing the genome size and content of close relatives with different life strategies. So what I've done here is plotted a free-living Burkholderia on the far left, a facultative fungal endosymbiont, and then 2 obligate fungal endosymbionts here. So this is the study target, Mycoavidus, and then along the bottom you have genome size, and these pie charts are gene ontology or GO categories. So a couple of takeaways, as these bacteria transition from free living to facultative to obligate endosymbiosis, not only do their genomes physically shrink, they're losing genes, but they also change in the relative proportion of functional gene content. And we think that's a response to the selective pressures that these bacteria might encounter on their own, free living in the soil versus when they enter a eukaryotic cell. So we can use that genomic sequence to make predictions about the relationship between the 2. Here I've illustrated the bacterial genome content with the transporters around the outside, and then various aspects of metabolism in the middle. And focus with me just for a moment on primary metabolism. So here I've coded anything in blue as a gene product or a pathway that's impacted by loss or inactivation. And so I just point a couple of things out. The first thing is that 1/3 of the amino acids in this genome are missing, right? You need amino acids to build proteins. Proteins are essential for life. So these are certainly products that come from the host. And we can sort of reinforce this logic by looking at the transportome. So for example, we have histidine missing. Here's an importer specifically for histidine. So we've used this logic to try to figure out what's missing, what can this thing physically bring in. Those are resources that come directly from the host. And the other thing that's interesting here is that glycolysis, or the ability to drive acetyl-CoA from sugars, this is abridged in 3 separate places. So acetyl-CoA is really necessary for the TCA cycle and other cellular processes. And in this microbe, it's coming directly from beta oxidation or the breakdown of fatty acids. So even though this genome is super streamlined, it's lost a lot of genes, very small, it has multiple genes that code for enzymes in this pathway, as well as importers for fatty acids. So these amino acids and fatty acids are certainly products that come directly from the host. And this is cool evolutionarily. It gives us a time point on the far end of that spectrum, like how are symbioses that are very old and ancient. But you might guess that experimentally it's quite challenging to study something that you can't culture because it can't live alone. So what we did instead was take a clearing approach where we passage these isolates with multiple antibiotics back and forth on solid media and liquid media until we got negative results with our custom 16S primers. And what results then are 2 isolates in the same genomic background. So wild type on the bottom there has the bacteria living inside the cells, and the cleared line on the top does not. That's the same fungus with and without the bacteria. So qualitatively, you can see these strains are really different. The cleared strain is looking much better. And then quantitatively, we ask with the radial growth assay, how fast are these growing? That the cleared strain actually grows more rapidly. So we think in this condition, there's actually a fitness cost to hosting these bacteria, and that those resources that it uses, fatty acids, amino acids, et cetera, are redirected to other processes like growth and potentially toggling metabolism. And the other thing that we noticed when we got these isolates is that they smell quantitatively different. So you have them growing on plates, you take off the parafilm, you're passaging them, and you get this wet dog smell. So one is wet dog, one is slightly less wet dog. And this is not the first time that this fungus has been accused of smelling this way, which it does. So it was described in the late 1800s as garlic, cabbage, and wet dog. And what that actually is, you're picking up intermediates of that beta oxidation or fatty acid breakdown process. We have any pet owners, dog people? Yeah? Yeah? Yeah. So you know the wet dog smell. So what's happening there is that when you get your dog wet, you're picking up, so he's got oils on his skin, and the bacteria on the skin are breaking those oils down, so you're volatilizing those intermediates. It's the same thing that's happening. And so we smelled the wild type isolates, and they smelled more wet dog than the cleared, and we thought, this is funny. Let's see if we can put some numbers on this. So we sent our isolates off to a mass spectrometrist at Berkeley. This is out of Alan Goldstein's lab, and he uses a sniffer that's a bit more quantitative. So here what I'm showing you are mass to charge ratios. So each peak is an individual product, and a normalized quantification. And then the wild type is in blue, and the cleared isolate profile is in white. And I slightly offset them so that anywhere you see a difference in the height of the peaks is a product that's differentially abundant in the cleared and uncleared isolates. And then the products that are consistent with beta oxidation or fatty acid breakdown are in yellow. So in other words, when the bacteria are present, there's more breakdown happening. So this sniffer and that sniffer are in consensus. We've also layered on metabolomic data from Tim Chaplinsky's lab, and here I'm showing fold change of metabolites. So metabolites across the X, fold change on the Y, and I've organized them by category. So a couple things pop out. First of all, in the cleared strain, fatty acids accumulate. This is the class that's the most impacted. But also, they accumulate up to 8-fold. So in the absence of these bacteria, we're seeing an accumulation of the products that they use. But we hadn't quite nailed down whether or not this interaction was direct or indirect. So these bacteria are pretty clearly using fungal fatty acids. But what is the response of the fungi to the presence of the bacteria? We've taken a comparative transcriptomic approach to ask this question, quantifying mRNA in the cleared and uncleared isolates. And here I'm showing you the categories of genes that are impacted. So first of all, lipid and carbohydrate metabolism, which is great because this fatty acid phenotype keeps popping up in all our assays. And what I've done here is plotted the Saccharomyces orthologs onto a filament and then overlaid relative expression. Green is up and red is down, and I put a few colored boxes around metabolic processes to try to unbusy this a little bit. So the big picture is that when the endosymbiont is absent, we see less glycolysis and more aspects of some parts of lipid biology. So we think there's this metabolic toggle, and I'll remind you that this is anti-correlated with the capacity that the endosymbiont has. The endosymbiont doesn't do glycolysis, but it loves beta oxidation. So no sugars, but yes, fatty acids. Those are products that the host has to use and regulate, so you can imagine a sort of toggling of metabolism in response to these endosymbiont that we can see in the transcriptome and its presence and absence. So in conclusion, around plant roots and inside plant tissues, fungi and bacteria coexist and have the chance to co-evolve sometimes into these ancient symbioses like this. And I also think that Any microbe that is positive or mutualistic with a plant host that has an indirect, strong effect like this with another microbe actually has large implications for above-ground plant health. So with that, I thank my funding resources and had some awesome collaborators, and thank you for your attention. Take any questions if we have time.